Hello everyone, can you hear me well? Thank you. So at 3 now, a very good a very good afternoon to all of you who have joined us here. Welcome to the INBIOSIS webinar lecture series 2 this year. So I'm Dr. 𝐓𝐢𝐿𝐤, research fellow in INBIOSIS. So I will be your moderator for today's webinar. So I hope the video that we have played just now have provided some background of Institute of Systems Biology INBIOSIS, which located in University Kebangsaan Malaysia, Bangi, as a Selangor here. For those who are INBIOSIS new to you, I hope that the video, the sharing have provided you some insights about us, about our research, and also our institutes. So for those who are interested to collaborate with us or to know us more, please welcome to contact us. And you also can browse our social media to know more about our activities. So thank you again to join this today webinar. I'm glad to have all of you here. Salam Ramadan and a happy fasting month. This is a happy fasting month to our Muslim audience. So this is our second webinar of this year, and we will have more coming. And I hope all of you will join us again in the coming future after today's event as well. It is the time for me to introduce our honorable speaker, Dr. Law Chen Fei, a research fellow in INBIOSIS. So let me provide you some background about Dr. Law. So Dr. Law is a senior lecturer at the Institute of System Biology, also a research fellow here in UKM. He holds a PhD degree in molecular biotechnology from the University Putra Malaysia, UPM. His research focus mainly in aquatic animal health, including disease surveillance, host pathogen interactions, therapy, control, and prevention of disease. He applies systems biology approach, particularly competitive proteomics, in the study to comprehend the effects of prophylactic and therapeutic agent in the aquatic animals, as well as the host pathogen interactions. So his team also involved in a vaccine development using in silico approach and synthetic biology in vaccine production using 𝐈𝐭 as a host. So without further ado, please join me to welcome Dr. Law Chen Fei to share his talk with us today with the title Quorum Sensing in the Crosshairs, Targeting LuxP of Vibrio parahaemolyticus with Aptamers for Shrimp Health. Welcome, Dr. Law.
Thank you, Dr. Leong. Thanks for the very kind introduction.
It's yours.
Okay, sure. So allow me to share my screen first. Okay. So can you see the slide?
Yes.
All right, sure. Thanks. Thank you, Dr. Leong, for the introduction. So without further ado, I would like to start my presentation for today, which will mainly focusing on the quorum sensing in the crosshairs, targeting the LuxP of Vibrio parahaemolyticus with Aptamers for shrimp health. So this will be the main focus of my presentation for today. So I would like to start with a very brief introduction about the Aptamer molecules. It is actually oligonucleotides of either DNA or RNA molecules, a single-stranded molecule, where it can actually fold into a very unique three-dimensional structure. And then this molecule would have binding affinity towards a very specific target. So we can develop the Aptamers molecule against our target of interest. So among the advantages of the Aptamer molecules for clinical application includes that the molecule itself is non-immunogenic and it has a high penetration rate. So as I mentioned before, it can target on any target of interest, either protein or even metabolites. So in terms of the production of the Aptamer molecules, it is also quite easy to produce and the molecules is thermostable and it has less batch-to-batch variation. This is very important because the Aptamer molecule is sequence specific. So regardless which batch of production, it will still have a very specific sequence. So it has no variation between production batch. So the production also is cost effective and it requires only a short period of time. So in this research, we try to apply the Aptamers technology to target to sort of manage one of the frequently outbreak disease in the shrimp aquaculture, which is the early mortality syndrome, or also known as the acute hepatopancreatic necrosis disease. And this disease is affecting the shrimp aquaculture across the globe and including the listed here some of the major shrimp producer countries. So this is the clinical presentation of the AHPND, where the shrimp were presented empty stomach, pale atrophied hepatopancreas, and also empty guts, as compared to the healthy shrimp with full stomach, brownish hepatopancreas, and also full gut. So according to the World Organization for Animal Health, the Manual of Diagnostic Tests for Aquatic Animals, so far there is no vaccination applicable to tackle the disease, and also there's no available chemotherapy and also blocking agent. So there is no reported immunostimulants that are effective to manage the disease, and also breeding the resistant shrimp is not applicable. So the culprit of the AHPND has been identified as Vibrio parahaemolyticus, where the mortality can goes up to 100% in the culture stream within a week of the in the infected form. So the common pathogenicity of this bacteria includes the production of several soluble toxins that is known as the tox A and tox B, the most stable direct hemolysin and the DDH related hemolysin, and also two of the secretion systems, the type 3 and type 6 secretion systems. So for the AHPND, it is actually caused by a very specific virulent strain of this bacteria, which the strain contains a 70 kilobase pair plasmid with genes that encode for the homologs of Photorhabdus insect-related binary toxin, known as the PIL-A and PIL-B. However, removal of this plasmid actually can abolish the AHPND causing ability of this virulent strain. So we propose mitigation strategies to control the AHPND disease in shrimp aquaculture by downregulation of the virulent genes expression in the bacteria. So this mitigation strategies is feasible due to the fact that a lot of the virulence factor of the bacteria is regulated through the bacterial current sensing. And this is actually a cell-to-cell communication that coordinates the pathogenic behavior of the bacteria. So different category of the bacteria will produce different signaling molecules used in the current sensing. For example, here the Vibrio, our target bacteria, is producing the ferranosyl borate diester known as the autoinducer 2, or in short, the AI2 molecules, the signal molecule. For the current sensing, it is actually a cell density dependent mechanism, where increase in the concentration as a function of cell density. For example, here, at low cell density of the bacteria, it will produce low amount of signaling molecule. So as the bacteria population grows into high cell density, so it will produce more signaling molecules, and this signaling molecule will actually signal back the bacteria to differentially regulate some of the major gene expression that includes the virulence of the bacteria, the mortality and swarming of the bacteria, efflux plants, and so on. So knowing that the the objective of our study here is to target the LuxP receptor protein, which is a periplasmic protein that's sensing the autoinducer 2. So this is the receptor protein. Upon sensing the signaling molecule, so it will actually activate the phosphatase of this complex, the LuxPQ complex, which will actually dephosphorylate the Lux protein and leads to the repression of the AHPA. So this gene is actually the master regulator that controls the individual behaviors of the bacteria. So the repression of AHPA will lead to the expression of LuxAL and also or the HAPL, which is the master regulator that control the group behavior of the bacteria. So we tried the in silico approach to design the aptamers that can actually interfere with our target protein, the LuxP. So we constructed a virtual library of aptamers with a random sequence here and perform a molecular docking against the target receptor protein to select the potential candidates which can bind to the LuxP receptor protein. So from the molecular docking, we able to identify top 6 of the potential candidates as listed here with different affinity towards the LuxP receptor protein, and the VPL130 actually showing the highest affinity, where the binding affinity is the lowest at -9.2 kilocalories per mole. So following, we assess the interaction and also the stability of this complex, the aptamer protein complex, by using a molecular dynamic simulation. So, and also from the simulation, we recalculate the binding affinity using a molecular mechanics Poisson-Boltzmann surface area approach. So from our MD simulation analysis, we able to see that our complex is actually quite stable in terms of the, as you can see from the fluctuation of the protein backbone here, it is within the acceptable range of 0.2 nanometer. But we actually observe quite a significant fluctuation of the aptamer molecules, and we speculate that this mainly is caused by the linear structure of the aptamer molecules itself, because of the 3' and 5' ends that are not stabilized. So because of this, we propose several modification on the aptamer molecules prior to in vitro assay, so which I will be explaining in the later slide. So from the molecular dynamic simulation, also we study the hydrogen bond interaction between the LuxP receptor protein and also the aptamers molecule. So from here, we can see that the VPL110 actually forming the highest number of hydrogen bonds with our target protein. But I would say all of the 6 candidates are actually quite stable based on the hydrogen bonding interaction and also the RMSD of the protein and also the protein complex with the aptamers. Then we recalculate the binding affinity using the MMPBSA approach from the 50 nanosecond simulation time. So this video is showing the dynamics of the protein aptamers complex across 50 nanosecond. And from the recalculation of the binding energy, you can see that VPL900 actually having the lowest binding energy. But overall, all six candidates are actually showing quite promising interaction, which we can consider significant because of the binding energy is as low as -250 kilojoule per mole and below. So until this part is still so-called the in silico design is all based on computational analysis and prediction. So we further validate the prediction by in vitro assay. So this is the approach that we try to validate the interaction by using the isothermal titration calorimetry. And this is one of the very high sensitive instrument that is commonly used for the biomolecular binding study or biomolecular interaction. So with this ITC, we actually analyze the sample in solution form, which is quite close to the live sample that we will be using. And the nano ITC actually requires less sample because of the scale itself. And it's very sensitive because it can detect or record very subtle heat changes across a 20 titration of our ligands into the target protein. So in order for us to run the ITC, we need to obtain the LuxP receptor protein, and then that's why we perform the molecular cloning and expression. So we successfully clone the LuxP receptor protein into the E. coli hosts and express it. Then we able to purify the LuxP receptor protein, and we confirm the identity through protein sequencing, and all the peptide match with our target reference in the library with 76.4% of the sequence coverage. So we can very sure that our recombinant protein is actually the target LuxP. So this is the modification of the aptamers that I mentioned before due to the fluctuation. So we try to stabilize the structure by adding a GC complement sequence at the 5' and 3' ends here, so that it forms a self-complement ends with hairpin lobes here. This is a kind of graphical presentation. So this is basically how the structure looks like. So further, we analyze all the 6 potential candidates, but this is the only 2 that's showing a very significant binding, where the dissociation constant, the KD value, recorded at 0.2 and 0.5 micromolar. So at the micromolar level, so these 2 are considered the best binder to the LuxP receptor protein. The data output from the ITC is actually showing the thermodynamics of the interaction. So we can see the delta H, delta S, and delta G, which is the changes in enthalpy. For delta H and delta S is the changes in entropy, and the changes in Gibbs free energy. However, for the common or for the molecular interaction, the delta S, which is changes in the entropy, a decrease in these energy changes is actually not so favorable for biomolecular interaction. But in our case, this decrease in the entropy mainly we speculate that it is not dominantly caused by the decrease in the conformational entropy in the bound state, because we know that the LuxP receptor protein actually will undergo major conformational changes upon endogenous ligand binding. So showing here in the picture is two structure of the LuxP receptor protein, which is the upper form of the LuxP. You can see the opening of the structure before the endogenous ligand binds into the protein. And this is the hollow form of the structure where the endogenous ligand has bound in the binding pocket. So the animation also showed clearly that the structural changes or the conformational changes of the protein due to the endogenous ligand bindings. So, however, the decrease of the entropy energy here is actually compensated by the changes in the enthalpy, which is the delta H. So that's why we still can get a negative delta G, the Gibbs free energy, and this negative value indicates that the interaction between the aptamers and also our target LuxP protein happens spontaneously. So this is this actually tells us or confirm that our design aptamers is interacting with the target LuxP receptor protein. So since we have validated the interaction from using the ITC, so then we move on to validate the potential bioactivities of the design aptamer. Here we conducted a preliminary analysis, which it demonstrated a significant reduction in bioluminescence of a model organism, which is the Vibrio camberley. So this is the reporter of bioluminescence, where we know that the bioluminescence is actually regulated through the current sensing cascade. So by treating the bacteria culture, we see a significant reduction in terms of the bioluminescence of the bacteria in a dose-dependent manner. So that's why it also indicates that the aptamers that we have designed from the in silico approach is showing the bioactivity in terms of the anti-current sensing. And then we further analyze the bioactivity by conducting a comparative transcriptome analysis, where we treated the bacteria culture with 2 different concentrations of the aptamers at 10 micromolar and 100 micromolar. So we only tested on the best binder, which is the VPR 300. And also we included 2 different control, which is the non-treated negative control and also a positive control by treating the culture with furanon at 10 micromolar. This is the compound that has been reported to possess antiquine sensing activity. However, this compound cannot be used in the industry or in live animal due to its toxicity. So we only include here as a positive control to compare the effect of our Aptamer treatments. So from the comparative transcriptome analysis, we're able to see a different significant number of genes being differentially regulated upon different concentration of Aptamers treatment at 10 micromolar and 100 micromolar. So looking at this, the number of significant genes that is being differentially regulated here, we also can tells that the Aptamers treatment are actually in a dose-dependent manner. So by increasing the concentration of the Aptamer treatment, we can see more effect out of the treatment. So as compared to the positive control of furanon, this is a very potent compound. So that's why we can see a very significant number of differentially regulated genes. So after we identified the DEG, so we further with the gene set enrichment analysis to see the different pathway or different mechanisms being up or down regulated. So from the gene set enrichment analysis here, we can see that all the treatments actually suppress the current sensing related genes expression. And in addition to 100 micromolar treatments of the VPL 300 Aptamers here, we can see the flagella assembly related genes also being suppressed. So this is the details of the different genes involved in the current sensing and also the flagella assembly mechanism. And we can see all these genes are significantly downregulated or suppressed as compared to the control, the non-treated control. So particularly for the flagella assembly, also this is because we are expecting to see the suppression in current sensing related genes. But for the 100 micromolar treatments of the Aptamers, we also managed to see the flagella assembly, the suppression in flagella assembly related genes. And this is particularly important because the flagella also involved in the pathogenicity of the bacteria and in the parahaemolyticus, because they are switching between a streamer life cycle to a swarmer life cycle. As you can see from this, this paper, that explain comprehensively the switching of the life cycle of this bacteria, which the swimmer probably involved in the spreading of the bacteria, spreading of the disease. And when the bacteria attach to any surface or the host, then it will switch from the swimmer into a swarmer life cycle. And this is involved in the progression of the infection itself. So that's why by suppressing the flagella also, we hypothesize or we predict that the Aptamers treatment also could mitigate the progression of the infection in the animal. And at the same time, also, we tried another approach to select more potential candidates of Aptamers that can interact with the LuxP receptor protein by through this conventional method, which is a systematic evolution of ligands by exponential enrichment. In short, we call it SELECT. So this is basically the procedure or the cycle of the SELECT, where we start with a random library of different sequences here, and that they can form a very unique three-dimensional structure. So we select those structures that has a binding affinity towards our target by incubation. So those unbound Aptamers will be washed out and removed, but the bound, the Aptamers that binds to the target will be eluted out and amplified through PCR. So that is the function of we including the known sequence of primer at the 5' and 3' ends. So this primer will be used to amplify our library, and then we repeated the cycles to select until we get a high affinity binder of the Aptamers towards our target. So we perform this in vitro assay to select more potential candidates of the Aptamers that can interact with the LuxP receptor protein. So from the purified recombinant LuxP here, we perform the select by immobilizing the LuxP on the resin bit, and then we perform the selection. and across 14 cycle of the select. So from the select cycle of number 6, 10, and 14, we subjected these 3 cycles to next generation sequencing to obtain the Aptamer sequence from the library. So as you can see, this is the data from the NGS where where it recorded a decrease in the unique number of sequence across as the select cycle progress. So we're getting less and less unique sequence because we enrich several sequence throughout the select cycle. So this indicates the successful of the enrichment through the select cycle. And also the percentage of sequence duplication also increase across the selects as the selects progress. So these two data actually showing the progression of the enrichment throughout the select cycle. So here we obtain another 5 potential binder with the different percentage of enrichment. Of course, the sequence one is so far the highly enriched sequence. And we perform again the ITC to study the thermodynamics of the interaction and to look for to see or to examine whether or not this sequence is actually a better binder compared to our in silico design aptamers. So for sequence 1 and sequence 3, we can say that they are actually the better binder compared to the in silico design aptamers because it recorded the KD value at 23 to 24 nanomolar compared to our in silico design aptamer at 0.2 micromolar scale. So, however, we haven't managed to analyze on the bioactivity of these 2 molecules because the research is still in progress. So I can only update our research up to this stage lah. So if this is one of our study lah. So if I have a few more minutes to introduce another application of the aptamers also in related to stream health study. For this research, we actually focusing on the giant freshwater prawn, which is affected by the outbreak of Mycobacterium rosenbergi norovirus infection. So this is a disease that affecting the giant freshwater prawn, and the infected prawn will show the whitish post-larvae. So as you can see from the picture here, the opaque musculature of the affected prawn. So another name of this infection, this disease is also called the white muscle disease of the giant freshwater prawn, because the infected prawn will present a white muscle. And the mortality is also quite high. You can observe the mortality from 2 to 3 days post-infection, and the cumulative mortality can goes up to 95 to 100% within 5 days. And showing here is actually the virus particle. So it is the mRNA, the virus is actually a non-enveloped virus. So it consists of 180 subunit of the capsid protein, which encapsulates the RNA genome of the virus. So we directly target the capsid protein of this virus, and we select the potential binder of the aptamers that can bind to the capsid protein. So similar to the previous study, we clone and express the mRNA capsid protein in E. coli, and we successfully purified it and confirmed the identity through protein sequencing, and showing here the coverage, the sequence coverage of the sequence peptide against the reference from the library. So we confirm the identity of our target protein, the capsid protein here. Then we perform the select cycle again by immobilizing the capsid protein on a resin bead, and then undergoes 12 cycles of select to identify the potential aptamers that can bind to the capsid protein. So we subjected three libraries of the aptamers also from cycle 4, 9, and 12 to next generation sequencing. And similar to the previous study, we can see the enrichment by from here through the decrease in the unique sequence obtained across the cycle, and also the increase in percentage of sequence duplication. So meaning that several specific sequence are being enriched across the select cycle. Okay, so showing here is the top 5 candidates that can bind that can bind to the capsid protein. But in this case, we have a different application in terms instead of looking at the bioactivity of the aptamers, we apply the aptamers candidate in developing a gold nanoparticle-based apta sensor to detect the viral protein. So basically, this is how it works from the gold nanoparticles. We coated the particle with our aptamer candidates, and in the presence of sodium chloride, so the coated gold particle will remain dispersed in the solution. But in the presence of the viral protein, the aptamers, which has a higher binding affinity towards the viral protein, will actually bind to the viral protein. And in the presence of the sodium chloride, the nanoparticle will aggregate and it will change in color. So as you can see from this the microtiter plate here, so here we determine the minimal concentration of the sodium chloride that can cause aggregation of the gold nanoparticle. So we use 30 millimolar of sodium chloride in our test result, in our test sample, to see to examine the sensitivity of these aptamer molecules in detecting the viral protein. So from the top 5 binder, so we tested only, we tested, because the research is still in progress. So at this time, we only managed to test 2 of the aptamer candidates, which is the CPA1 and CPA3, where the CPA1 actually showing a higher sensitivity because it can detect as low as 62 nanomolar of the viral protein compared to the aptamer CPA3, where the detection limits is actually only down to 250 nanomolar. So we're actually interested to develop this assay into a colorimetric assay so that it is easier to apply in the industry by just observing through using our naked eyes. We can see the color changes, then we able to justify whether or not the viral protein is actually present in our sample. However, for the detection assay, we need to, of course, we need to determine also the sensitivity and the specificity of the assay. But this is only our preliminary study. We just able to conduct the research up to this stage at this moment. So of course, in the future, we're going to test for the sensitivity of this assay. And basically, that is the research that we have done so far in terms of applying the Aptamers technology in managing the stream health in aquaculture. So to summarize, for today's, we managed to identify 2 potential endocrine sensing aptamers from in silico modeling approach to target the LuxP receptor protein from the parahaemolyticus. And then the in vitro assay showing promising bioactivity of this aptamer in suppressing the current sensing of the bacteria. And then the further detailed analysis in terms of the gene expression, we're able to show the suppression of current sensing related and also the flagella assembly related gene expression upon the aptamer treatments. And then for the selection of more potential aptamer candidates using the SELECT protocol, we're able to identify a few more aptamer candidates that showing higher binding affinity to the LuxP receptor protein. And hopefully in the future, in our future study, this potential candidate showing higher potency in terms of the endocrine sensing activity. So however, at this moment, we haven't tested the bioactivity of this aptamer in torrent quenching. So another application of the aptamers in the viral detection, the viral protein detection, which is targeting the norovirus here, we able to isolate or identify 2 potential aptamers that can be used to be developed into a gold nanoparticle-based apta sensor for viral detection. But as I mentioned before, we need to evaluate the sensitivity and also the specificity of this assay in the future. And so for the acknowledgement, would like to acknowledge the funder from Ministry of Science, Technology and Innovation, and also Ministry of Higher Education, and also the funding from our collaborators, Associate Prof. Dr. Natra Fatin from UPM, who has received the International Development Research Center fund from the Canada. And also the scientists who has done all the hard work in the lab, Noor Afika, Muhammad Yusuf, and also Noor Zigatin. Before I end my session today, so I would like to announce, I would like to share also, we're going to organize a Asian Regional Conference on Systems Biology at Swiss Garden Hotel, Malacca, from 20th to 21st December, end of this year, 2023. So the theme for our conference this year is Modern Biotechnology for Sustainable Development Goals. So please come and join us in the conference. So any further information, you can actually get it from our website here. So you can visit lcsb2023.wibly.com for more details information about our conference. And with that, I would like to end my presentation here. Thank you.
Thank you very much, Dr. Law, for the very insightful talks. Introduce us about the problem that we are facing in the shrimp industry with the LuxP protein, which is a quantum sensing for the Vibrio bacteria, and how the application of the Aptamers towards this controlling or anti the quantum sensing of this Vibrio bacteria, and also the virus that infecting the shrimp as well, MR and V. So I would like to open the floor for the questions, and while we are waiting, here you already in the chat already have questions from Dr. Mooney, so I will read out from here. So there's first question from Dr. Mooney. Is there any side effects to those who consume the animal with Aptamer plus vaccine?
Plus vaccine?
Aptamer as a vaccine, right?
But in our case, in this study, we are not using it as a vaccine. It's actually a sort of a therapeutic agent, I would say, because we try to utilize the Aptamers to suppress the gene expression in the bacteria. So it sort of will, we actually hypothesize that it will mitigate the infection. It will actually reduce the infection, or at least the progression of the infection. So it's sort of, I'm not sure whether I can use the term to treat the infection, because we are not killing the pathogen, we are not killing the bacteria. So we just suppress the virulent genes expression so that because the Vibrio is actually a commensal, it's an opportunistic pathogen. So in many cases, they will not cause infection. But if the fish is, or the shrimp is under stress, then the potential outbreak of the infection would occur. So they are kind of opportunistic. So by applying the Aptamers technology here, we hope to sort of limit the infection, because a lot of time the infection is actually caused by the virulence factor due to the toxin that the pathogen actually secreted. And then yeah, so but in terms of the safety, I would say it is safe, because the Aptamer molecules, as I mentioned, because previously, in our case, we are actually using the DNA Aptamer, so it is a biomolecules. So I suppose they will have what do you call it? Less toxicity lah, because it's a normal DNA molecules. It's just that it is a very short sequence and a single-stranded DNA molecules that fold into a unique three-dimensional structure. So I believe it is less toxic, but we will, of course, we still need to test for the toxicity lah, if we're going to apply it in the animals and also for consumption later on. So that is the further study that we're going to conduct in the future. But at this point, we only showing the potential of applying this technology in the treatment, because apart from the animal study, actually the Aptamers technology has been widely applied in a lot of human research that includes cancer study and also several infection study, whether or not the pathogen is from bacteria or virus. And I would say it has less toxicity because there is already one Aptamers product in the market, which is 𝐈𝐭 The Aptamers has been used to treat the neurodegeneration of the eye nerves in human. So if I'm not mistaken, but I'm sure that the 𝐈𝐭 is actually an Aptamer-based therapeutic agent that used in treating human disease with the neuron degeneration of the eye nerves. Yeah, so that's why I would say I'm quite confident that actually we will see less toxic effect from the Aptamers treatment.
All right. I actually having a similar question, like Prof. Michael Wong here. So the question was, how stable is the Aptamer, and or how long is effect in say a swim pond? And also in addition from this question in my mind also, how because when we are talking about applying on the pond, and how much that you think or any idea of or sharing on how much we really can practice of putting how much of DNA in the pond, for example. So there's three questions here. One, how stable is the Aptamer if we put it in the environment? And how
Okay.
long is the effects? And the third is probably the amount that we need to apply into the pond, perhaps.
Okay. I'll start with the last question first. In terms of the amount, if we're going to apply it in the pond or in the tank, or directly apply it in the water body of the aquaculture system, I would say that will require a lot of it will require a very high amount of the Aptamers itself. So that's why we actually not targeting to put the Aptamers in the water. Actually, we actually aims to incorporate the Aptamers into the feed. So that's why we will use we target to use less Aptamers in terms of the amount when we incorporate into the feed and directly feed the stream. So that is one of our target lah, to develop the AptaFeed for the treatment. And the next question on how stable it is, the Aptamer molecules in the system, we haven't really studied that yet. So, but we expect to do modification of the Aptamer's molecule to increase its stability and also the bioavailability, because even for the Aptamer's products that has been commercialized in treating the eye disease, they did perform modification, and a lot of research has been done. One of the easier way to stabilize the Aptamers to increase its stability and also the bioavailability when it is in the animal system is by incorporating the PEG, the polyethylene glycol. So it is a polymer that can stabilize the Aptamers molecule and also increase its lifespan in the animal system. Yeah, so of course, at this point, we're only analyzing on the potential bioactivity of the Aptamers to treat to suppress the gene expression in the bacteria. We haven't go into the animal study yet, but we expect to we expect to do further modification lah, in the sense to increase the stability. So, but that is not yet covered in the current research here. Yeah.
All right. Thank you for the sharing. And there's another question from Dr. Mooney. Is that true that antibiotic cannot get rid of the quorum sensing in bacteria?
I'm not aware if there's any antibiotics that directly targeting the quorum sensing, but as we all know, the antibiotics is applied to kill the bacteria, to actually treat the bacterial infection. So it's either those antibiotics either are bactericidal or bacteriostatic. But even though there's no specific treatments or prevention of the AHPND in shrimp aquaculture yet, but at this point, farmers are actually applying a lot of antibiotics. So the application of antibiotics in aquaculture is actually very common. But because of that, we actually need to come up with a more strict regulation in terms of antibiotic use, because recently it has been an issue of the emerging of the antibiotic resistant strain from aquaculture. So there's a lot of disease outbreak in shrimp and also fish aquaculture, where we can see we able to isolate a lot of bacteria and tested with a range of antibiotics, and they are actually resistant. So this is getting serious, and that is why the IDLC, the International Development Research Center from Canada, offer the call for proposal to look for alternative instead of using antibiotics. So to look for alternative potential treatments and also disease management, so that we can reduce the use of antibiotics. And at the same time, also there are a lot of study being conducted to evaluate the emerging threats of this antibiotic resistant bacteria in aquaculture, because it's quite serious problem when we apply the antibiotics in the aquaculture, because it's directly to the environment and to the water body, and it can actually spread to the land as well, because of the water as the carrier. So that's why, yeah, I hope I answered the question. Actually, I forgot the core of the question is asking.
Yeah, I think antibiotic probably pretty, if you apply, then you training all other bacteria, right, towards
Yeah,
that.
yeah.
For the resistance is pretty bad, right? All
So
right,
that's why
I
this
think
is okay.
yeah, we have more questions, but I think couple of questions have been we have asked similar question how to apply it to the large population of pond. Dr. Moni, Dr. Maizum, I think we are having similar question before, and Dr. Low also have addressed the question incorporated the Aptamer into the feed, right? So that's the question from Dr. Moni as well. And we have question from Al Hassan Osman. Are there any in vivo study with Aptamer technology? In vivo study with Aptamer technology? If yes, how stable is it? It means fitting the Aptamer into the animals, perhaps, right? It's like what you meant. You put the into the feed, and the animal will eat it, and it will still work in the animal. I think that's probably the questions.
Yeah, actually, there are studies showing the potential use of this Aptamers technology in vivo, because especially on I'm talking from the aquaculture side lah. So there's also a lot of study being done where the researcher actually identified and selected several potential Aptamer binder that can target the aquaculture viruses. So where they have tried a very small scale of animal study, and the results are very promising, where it can, by applying all these Aptamers, it can actually prevent the spreading and also the infection of the viruses. So that's why we see the potential in this. But from my limited reading, I haven't seen any large scale study. I mean, in terms of industrial scale study in the farm, the application in the farm. So a lot of publication and also a lot of report are actually at the lab scales. So it's still at the research scale, not yet to the industrial scale. So I would say it is still a promising approach in the therapeutic side and also in diagnostic side, because this is I would say this is the commonly application that people applying the Aptamers in lah. So to treat the disease as a therapeutic agent or to detect the disease or detect the pathogen from the diagnostic part. So all the laboratory results are showing the promising data that they can be applied here. So, but yeah, at the farm scale, I haven't come across yet lah. So I'm not sure whether or not if there's any industrial scale of study being conducted. And for myself, of course, I wish to go to that certain to go to that level to apply to try to apply in the industry, of course. So but we are still in progress. So hopefully in the next few years we're able to go to the industrial scale.
Right. So there's a question from Facebook. Will it have collateral effects on other beneficial microbes when applying the Aptamer?
Well, there's another concern as well because but I would say from my understanding, I would less concerned about this because it is not a very potent compound or a very potent agent here we're using, unless we're talking about antibiotics, because antibiotics we know that it will kill microbes. So it might also kill some of the beneficial microbes in the guts, regardless in the animals or in humans. So but in this case, we are actually developing an Aptamers that's specifically targeting the LuxP. And that LuxP is actually regulating the quorum sensing cascade. And the quorum sensing is involved in the regulation of the virulence of the bacteria. So I would say our target is very specific, that they might not have a lot of side effect on other beneficial bacteria in the host. I would say if from that perspective lah. Unless the Aptamer able to kill the bacteria, then and it is non-specific, then yeah, I would have that concern. But here, because the Aptamers molecules are known to have their specificity, because we are developing the Aptamers specifically targeting the LuxP only. And in the select cycle, so we did perform some negative selection. So that is to exclude some of the non-specific binders. However, of course, we can't cover all the potential non-specific binders. So we only select a few target for the negative selection. But still, through the enrichment of the affinity, based on the binding affinity, through the enrichment of the select cycles, we believe that the end product of the Aptamers is only specifically targeting the LuxP. So I would say it will have less effect on other beneficial bacteria. Yeah.
Right. Thanks for the sharing. So there's another question from Zarina Amin. How about using an Aptamer against AI2, seeing it's a universal cell signaling molecule? Any thought of this? VS LuxP, that means Vibrio LuxP?
Yeah. Yeah, from my understanding is that we try to neutralize the signaling molecule instead of targeting the LuxP, right? So there's actually another approach also. We can also
Versus
develop,
LuxP, sorry for that.
yeah, we can also target the AI2, and that will actually blocking the AI2 from being detected by the receptor protein. But however, we didn't go for that approach because from my opinion or from my perspective is that we don't know the amount of AI2 is being produced by the bacteria colony. It would be very high in concentration, so we might need more Aptamers to counter every single signal molecules. But compared to the number of protein that present on the bacterial surface, I would say it would be less than the AI2 molecules in the animals or in the culture media. So for that reason, I would say it is easier for us to target the receptor protein instead of the AI2. Of course, there are different study also being conducted targeting all those signaling molecule. One of it is that they have developed, they have identified an enzyme that can actually hydrolyze the signaling molecules. And this is also another approach to sort of interfere with the current sensing and suppressing all the virulence factor expression in the pathogen. Yeah, so that approach also possible. But because we're not able to do everything. So that's why we can only focusing here targeting the LuxP. But there's another nice approach also, I would say, by targeting the signaling molecule. We can try on that as well. Yeah.
Yeah, definitely. I think more room to explore over there.
Yeah.
Yes. Another question, could we use microbiome to see the effects? I suppose testing the microbiome by applying to Aptamer.
Meaning in vivo or in vitro? I suppose it's in vivo, right? The microbiome.
Yeah, suppose.
Yeah, of course. If we're interested to see whether or not by applying the Aptamers, if it's going to affect the microbiomes of the stream guards, so probably we can try that approach lah to study on the microbiome to see if there's any changes in terms of the microbe population in the guts. So that can help us to evaluate whether or not it will actually affect those beneficial bacteria as well. Yeah, that's one of the nice approach. Maybe we can try to consider in our study as well, because we're going to see also whether our Aptamers, when we apply to the animal, if there's any effect to the microbiomes of the stream. Yeah.
Right. So since there's no question from there, maybe I ask one question. I would like to ask on the bit of technical part over here. So you share with us about the selecting the Aptamer and then conducting the docking or simulation on selecting the species which is can bind tighter to in this case, the LuxP, right? So from the beginning of the species of the Aptamer, are there any, or I missed out something, or you given background on how actually began? Because Aptamer can be many of any of the nucleotide combination, right?
Yeah.
So are there a selected target already from the beginning to select? And the one that from the screening was the one that using later apply into a experiment that you use for the selection?
Okay. Yeah, here actually we started by constructing a very small Aptamers library, and the initial library is only 4 nucleotides actually. So we have all the random sequence in this library at the starting library of 4 nucleotides. So it actually consists of 356, if I'm not mistaken, the candidates. So this cover all the possible sequence, the random sequence of the library at the initial library. So from the 4 nucleotide sequence, and we selected those with the highest binding affinity, yeah. And from there we elongate the sequence. So meaning that from 4 nucleotides, we added 1 nucleotide at a time, and we perform the docking against to see whether or not it increase or decrease the binding. So that is actually quite tedious lah. It is quite time consuming, because we elongate it one by one nucleotide at a time. So until it reach at 8 nucleotides in one single molecules. So what we have presented here from for the dynamic simulation, we actually perform using the 8 nucleotide sequence. So all these selected, the 6 potential candidates are actually only 8 nucleotides long. So together with the modification of another addition of 8 nucleotides that complement at the 5' and 3' end, so the whole molecules itself is very small. It's only 16 nucleotides. Yeah.
Right.
So this is the
Yes.
Yeah, basically this is a molecule actually. So we predicted the molecules, the potential structure of the molecules. So the 5' and 3' ends are actually self-complement. So it's forming a hairpin loop. So here consists of 8 random nucleotides where we selected from the docking and also the simulation. So these are the nucleotides that in the loops. So it has the free base that can interact with our target protein.
Yep. So during the experiment, when you apply during the selection in the selects, so the design, because selects is random of the nucleotides, am I right? In this case, so is that the selected nucleotides from the selects or selected aptamer align with from what is predicted, especially in these 8 nucleotides?
For that one, we haven't studied yet. Yeah, we haven't examined that at this moment. But as you can see from here, where was this? Yeah, this one. This is actually the S1 and S3 sequence. I purposely make it blur because it has the potential for future application. So that's why I couldn't review the sequence yet at this moment. So that's why I blur the sequence. So you can see this is the 2 predicted structure compared to our in silico design one. Is very different in terms of size, and also all these nucleotides that, especially in the loops, in the loops here, they are they are the potential nucleotides that can interact with the protein because they have the free bases for the interaction. And also another thing is we are yet to know which part of this molecule are actually interacting with the binding pockets, because it can also bind to the surface of the protein at another side instead of the binding pockets. So we are not sure which part of it is actually in the binding pockets or interfering with the binding pockets. So that is also in my mind actually to conduct the study to see whether or not it actually cross between the in silico selection and also the in vitro selection. So if we manage to identify the what do you call it, the significant structure from here that is actually contributing to the binding affinity, we might wish to truncate the aptamers here, because the larger the molecule, of course, it will increase the cost for production. So that's why we might in the future also will truncate the aptamer so that it becomes smaller in terms of size. So it will require less cost for production, but at the same time, it has also a higher binding affinity. So yeah, there's actually a lot of study that we need to conduct in the future to get it optimum for the application in terms of production and also to the industry. Yeah.
That's right. So as a structural biologist, I also interested. It would be great if we can see where exactly the protein or aptamer is binding, right, to the
Yeah.
protein, and I think that will also provide validation to the uh, target that's selected from the MD or simulation, right?
Yeah, if we can identify the parts of aptamers that are not contributing to the binding, probably we can just truncate it out. We can just cut it out. So leaving just only the interacting part of the aptamers. That's
right.
only able to do when we have the maybe probably the crystal structure of the complex. Then only we can know which part of the aptamers are actually interacting or contributes to the binding. Yeah.
Yeah, that probably would help. Right, we are approaching 4:05 now, 4:04 now. Very hot discussion over here. Thank you for all the questions from everyone. And so thank you again for Dr. Law for sharing with us these very interesting topics.
Thank you. Thanks.
Also, I think get us to think more and explore more of the possibility of molecular biology as general, but aptamer more specifically. So so we are approaching the end with our seminar here. So I would like to thank again Dr. Law for the sharing.
Thank you.
Yeah, and I would like to also thanks all the participants over here that participating for all the questions and contributing all the questions for the discussions. And before we actually end, as usual, we would like to invite you, all our participants, to switch on your camera if possible, so that we can snapshot of nice photo of you.
Hi participants, kindly switch on the camera for photography session. Alright, ready? 1, 2, 3, smile. Okay, another one. 1, 2, 3, smile. Alright, thank you.
Thank you, everyone. Also, we will post the attendance link for today's webinar at the screen, as well as a chat box. So please fill in your attendance. It will be available only for 30 minutes. So after that, the link will be closed. So please stay safe, take care, everyone. Have a good weekend ahead. I look forward to see you again in our next coming webinar. Please feel free to visit us in INBIOSIS as well. Thank you. Bye for now.
Thank you. Thanks.